Hey guys, so in this video we're going to talk a little bit about lymphocytes, which are introduced here in this most recent lecture. And the reason is because lymphocytes are one of the biggest components of the immune response and the adaptive immune response, and are really important. Plus, I did my PhD on T cells, so I think they're pretty cool anyways. So I'm going to talk a lot about them. Okay, so we have two major subsets of lymphocytes. We have our B cells and our T cells. And I think the best way to do this is to compare them both. So they look very similar. Basically a round cell with a round nucleus and a little bit of cytoplasm. And but B cells, their main role is to produce antibody or immunoglobulins. And remember that B cells can actually have antibody bound to their surface, and we call that a B cell receptor. Or they can have immunoglobulin that's soluble and floating around, and we call those antibodies, abbreviated AB. T cells have membrane-bound receptors that we call T cell receptors. So these two cell types are similar in that respect, but they can't. Their T cell receptors aren't soluble, so they can't release them like antibodies like B cells do. You'll hear other B cells, the subset called plasma cells, and plasma cells are B cell lymphocytes that are basically just professional antibody producers. They're just really good at doing it, and that's their only job. So you'll hear the term plasma cells when we're talking about antibody production. T cells have two major subsets that we'll talk about, and we'll discuss some subtypes of those subsets as well. But for the most part, we'll talk about CD8 T cells, CD8 positive T cells, and CD4 positive T cells. And we'll draw a couple of distinctions between these two types of T cells. So CD8 T cells, let's go down here. CD8 T cells and CD4 T cells. CD8 T cells are actually what we call cytotoxic lymphocytes. Cyto meaning cellular, toxic meaning killing in this context here. So CD8 T cells are great at actually killing cells and killing microbes. So these are kind of the soldiers of the lymphocyte army here. Whereas CD4 positive T cells are also called helper cells. And what does helper cell mean? Well, it helps other cells do their jobs, and it actually is kind of like the maestro of the immune response in that it can kind of direct it one way or another. So CD4 T cells are important in orchestrating the immune response, whereas the CD8 positive T cells are kind of, again, your soldiers that go out there and do a lot of the heavy lifting and cell killing. So those are the two subsets of these T cells. The other important thing to keep in mind, which we'll talk about here in a bit, is that a major difference between these is in their MHC, or major histocompatibility complex, types that they recognize. And so in the next video we'll talk about MHC and how that's important to these T cell responses. So these are kind of the main differences. The other main difference between B cells and T cells are that B cells mature within the bone marrow. They're produced and mature in the bone marrow early on. Whereas T cells are produced in the bone marrow and then they go out and populate the thymus, where they become differentiated. And so this thymic education and differentiation of T cells is really important in the development of the immune response, and we talk about that in class as well. So hopefully this video helps a little bit understand the differences between B and T cell lymphocytes, and also the subsets of T cells. Okay, so we've talked about the different lymphocyte subsets, and we've also discussed in lecture as far as why it's important to have this wide spectrum or wide array of different epitopes that are recognized by lymphocytes. You know, we talk about how there's millions and millions and millions of different epitopes recognized by lymphocytes. So why is that important, and how does that occur? So let's say that we have 4 populations here of lymphocytes. Let's call them T cell lymphocytes, but these could be B cells. And the first one recognizes the epitope A. The next recognizes the epitope B. And it doesn't matter what this epitope is, but the next recognizes C. And then the fourth one recognizes D. So each one of these T cells recognize a different epitope. So they're unique. All four of these are unique. The blue one will not recognize B epitope, nor will it recognize C. So keep that in mind, and that's why it's important that all of these lymphocyte populations are unique. So, okay, we have this population, and this is what we call the repertoire of lymphocytes in an animal. It basically covers the entire list of epitopes that the lymphocytes can identify and respond to. So let's say that this dog with this repertoire of lymphocytes is infected by a microbe. Beep boop. Okay, is infected with a virus that expresses epitope B. So some part of the virus, whether it's the envelope or the nucleocapsid, it has this epitope that's recognized by one of your lymphocytes. So this is great. This means that you have lymphocytes that can recognize this virus and respond to it. Unfortunately, this means that these other three populations, A, C, and D, can't respond to it because they can't recognize this B epitope. And so what happens is that when the antigen is presented, when this B antigen is presented to the B-specific lymphocyte, this one gets activated and it starts multiplying. So it splits, and these further split, and these further split. And at the same time, every other epitope that's recognized, so maybe this does have D epitope, then this would start activating and proliferating. These start proliferating, and within a matter of days to weeks, you go from one cell that's specific for that epitope to millions and millions of cells by this proliferation. And so this is kind of that clonal selection theory where a single clone can expand to become a large number of these lymphocytes. And the reason this is important and helpful to the animal, to the dog in this case, is that these B protein specific or B epitope specific lymphocytes can attack this virus and result in it being destroyed. So if you don't have a B specific lymphocyte here, then you might not be even responding to this virus. The good thing is that because your body has such a huge repertoire of epitopes that it recognizes, usually there is at least one lymphocyte that can respond to that specific epitope. So you might have that lymphocyte that can respond to that epitope, but the question is, how will it ever know that that epitope is around? And so there's really two major ways that lymphocytes can actually see their specific epitope. So as you can imagine, there's only one cell that's specific for that B epitope, and it's sitting over in your popliteal lymph node, in the dog's popliteal lymph node, and the infection is on the nasal mucosa. So what happens? It's on the opposite side of the body from where that lymphocyte needs to be, and it needs to see that antigen and that epitope in order to activate and proliferate and help to protect the dog. So there's two major ways that that lymphocyte can eventually see that antigen. And then the first one is lymphocyte circulation and homing. Circulation and homing. And basically all of this means is that your lymphocytes are driving on the highway almost constantly. So and the highway is represented by your lymphatics. So your lymphatics and your vasculature. And so lymphocytes, they'll stay in lymph nodes for a little bit, but then they'll recirculate in the body. And the reason that they recirculate and go to different places, they might home to other different types of tissues, is so that they increase their chances of finding the epitope that they're specific for. So it's like their long lost love, and they just can't stop searching until they find it. So that's one of the major ways that lymphocytes end up seeing that epitope and being able to respond and proliferate. The other, and perhaps the more effective way, is through what we call antigen presenting cells. And these cells, we'll talk about antigen presentation through MHC to T cells. But these APCs, or antigen-presenting cells, include macrophages, primarily macrophages, and dendritic cells. And the great thing about these two cell types is that they like to eat up pieces and particles of microbes and other molecules. And in doing so, they pick them up, essentially eat them, and then they travel back to the lymph nodes where those T cells are residing, and they present them to the T cells, or the B cells. So they, the macrophages and dendritic cells might be in the nasal mucosa. They pick up that part of that virus, and they start shipping it around to lymph nodes. And eventually, as they're presenting it and showing it to different lymphocytes, they might hit a lymphocyte that actually recognizes that and starts getting activated and proliferated. And it's these two major ways that facilitate the ability of your lymphocytes to actually identify and respond to their specific epitopes.